only within this group. And what kind of the skin, the cell outer part of organs stratifies squamous epithelium is the skin of this group. This is the function for regulating suitable temperature for sperm because the spermatogenesis or the process for design the sperm cells require the lower 2 to 3 Celsius than the body temperature. Normal body temperature 36.5 to 37, right? But for the temperature that are suitable for the sperm, just 34 to 35. Because white testes move toward the scrotum out of your body. And the thermoregulation comes from three processes. The first process from the contraction of the glandulous muscle within the scrotum here. This one, deeper to the skin here, you can see the sphincter on the scrotum because the contraction of glandulous muscle to protect heat loss from the scrotum or testes. Okay. And the second called the counter-current heat exchange mechanism that comes from the release the heat from arteries because the artery comes from the body. The artery pass through the stomach cord. This area called the stomach cord. The artery comes from the body towards the scrotum. The temperature from artery 36.5 like that from your body, right? And in the stomach cord you can see here. Papineform venous plexus, the venous network. The venous network left around the artery here. Okay, and the heat transfer from the artery into the lower temperature within the vein. Okay. So when the artery offers alive the testes, the temperature of blood decreases to suitable for like 34 Celsius. Okay. It's the mechanism of the counter-current heat exchange mechanism. And the third one is the contraction of cremasteric muscle. The cremasteric muscle normally is wrapped around the somatic cord here. Okay. The cremasteric muscle will relax when the increased body temperature. Okay, and it contracts when the temperature outside the body decreases. And move to the testes. This is the whole testis cross section. Okay. You can see the outer tunica abuginea here, over here. Okay. And tunica vaginalis might be detached. Normally it's located here at the cross and here. The tunica vaginalis contains two layers. The outer layer called the parietal layer. This one is the parietal layer of tunica vaginalis, and this one is the visceral layer of tunica vaginalis. They contain egg between them. Okay. Both tunica vaginalis, parietal layer, and visceral layer is the simple squamous line with only one layer of squamous cell, called the simple squamous epithelium. And deeper to the tunica vaginalis, you can see the connective tissue is called the tunica albuginea. It's bigger than vaginalis. and deeper to tunica aeruginosa. The deeper part of tunica aeruginosa, you can see our numerous base cells located in the deeper part of aeruginosa is called the tunica vasculosa here. So there are three parts. The visceral layer and parietal layer of tunica vaginalis. Deeper to it called the tunica aeruginosa, containing connective tissue. And the deeper part of aeruginosa is called the tunica vasculosa, containing muscular vessels. Okay. And the one side of tunica aeruginosa, you can see the thickening part is called the mediastinum stasis. Here. Within the mediastinum stasis, you can see numerous ducts. This one. And this duct called the late testis. The late testis. Okay, within the area the name mediastinum. Okay, this slide shows the duct pathway. First, sperm are produced within the first duct called the seminiferous tube, the first of sperm are produced here. Okay. Numerous logo of the seminiferous tube here. Okay. The spermatogenesis occurs within these two. When sperm are produced within seminiferous tube, is moved to tubular recti. The tubular recti or tubular recti is a small or the transition, just only the transition zone between the seminiferous tube to the late testis, the third duct. The tubular recti located here. And when the sperm move through the pass through the tubular recti, it goes to the third one, late testis, that located within the mediastinum. And then sperm pass through the late testis and go through the efferent ductus. The efferent ductus is the connection between testis, the testis, and pass into the epididymis. And the five are called epididymis. Normally, the epididymis are divided into the head, body, and tail of the epididymis, but they contain the similar epithelium. And after the epididymis, and move through the vas deferens. This one, okay, vas deferens, and go into your body through the inguinal area. This is the pathway of this sperm. We move to the internal or component of testes. This slide is calculation that is too easy to clear, so to get it. This one is the testes. When we cross section the seminiferous tube, you will see the basement membrane located here or tube. Here is the basement membrane. Okay. Then, and we can see the modified smooth muscle cell here, the squamous cell. And this cell called the myoid cell. It's a function like a smooth muscle. It can contract to release the sperm to the lumen. Okay. This is the one component of base located on the basement membrane of seminiferous tube. And the one type of sperm called the spermatogonia. The spermatogonia just only line on or closely to the basement membrane. And the spermatogonia or spermatogonia. And spermatogonia differentiate to be the primary spermatocytes. Primary spermatocyte differentiate further to be the secondary spermatocyte, and the secondary differentiate to be the spermatid. There are two subtypes of spermatid: round or early spermatid, and the last stage of spermatid to be the elongate or late spermatid. Okay. Round spermatid is the sperm look like a circular shape and move to contain the nucleus, midpiece, and reticulum. This one called the elongate spermatid. Okay. And between these two, it's not only contain the spermatogenic cell, but it contains one major supporting cell that called the Sertoli cell. The Sertoli cell here. The Sertoli cell, generally the Sertoli cell located on the basement membrane, but every cell is run through and give the numerous process. And the apical surface of this cell lie closely to the lumen. Okay. But we can't see the plasma membrane of this cell. We just only see the pale stain of nucleus of this cell. And in the test tip, you can see this one is the seminiferal tubule here, and this one is the seminiferal tubule here. But there are the space between tubule here, and this space called the interstitial space. The interstitial space contains the blood vessel and the one important cell that you have to learn today, Leydig cell. The Leydig cell located between the interstitial space between tubule, and the main function of Leydig cell secrete male hormone testosterone. Okay, we move on to the next slide. Okay. And the real picture of this light microscope. You can see this one, the squamous cell right here. This one is the myeloid cell. And this one, the dark stain of the nucleus located in the basement membrane is called the spermatogonial cell. Just only the dark stain and not too big nucleus. Okay, like here for the spermatogonial cell. And the spermatogonia. And the spermatogonia divide to the mitotic division to be the primary spermatocyte. Okay. You can see the numerous dots within the nucleus of this cell is the special characteristic of this cell, and the nucleus bigger than spermatogonia. Actually, the primary spermatocyte contains the biggest nucleus when compared with the outer type of spermatogenic cell. And after that, the primary spermatocyte divide the meiosis or division into the secondary spermatocyte. The secondary spermatocyte located nearly the lumen than the primary spermatocyte, and it dark stain nucleus than the primary spermatocyte. Okay. And then the secondary spermatocyte differentiate meiosis II to be the round spermatid here. The smaller size of dark stain nucleus of the round spermatid compared with the secondary spermatocyte. And some too, we cannot see two different types of the dark stain nucleus, merely the lumen. So we can assume it. They are the round spermatid than the secondary spermatocyte because secondary spermatocyte normally move quickly to be the spermatid. And this area show the interstitial space between the two groups. You can see the one cell. This one, the oval shape of cell. You cannot see the shape of cell clearly, just only see the nucleus. The nucleus, you can see the quite clear the nucleolus like here. Okay. And normally in the cytoplasm of each cell is a Leydig cell. The cytoplasm of each cell normally is stained with the red color, red or yellow, red or orange color. And the type of epithelium within the semi-infrared tubule, we just only call them the stratified epithelium because the stratified epithelium that means the more than 2 layers of epithelium. Okay, but we cannot identify specific type of cell because there are only one type of cell within these two. And I conclude, in this semi-infrared tubule contain only 2 cells, the spermatogenic cell and the Sertoli cell. We move to the information about the Sertoli cell. Okay. And this Sertoli cell, we just only see the pale stem nucleus of them. We can see the shape of this cell. Okay. And the irregular shape of nucleus are generally seen in the Sertoli cell. Sometimes the nucleus of Sertoli cell locate near or locate on the basement membrane. It's okay if you see the pale stem nucleus and quite bigger than the primary spermatocyte. Okay. And because of this cell locate on the basement membrane and its gives the nucleus process to bind with the other type of the spermatogenic cell because the function of Sertoli cell is to support everything for the spermatogenic cell. Okay. Between 2 Sertoli cells, Sertoli cell 1 and Sertoli cell 2. Okay, yeah. Between 2 cells connect to each other with a tight junction like here. The Sertoli cell form the drug-tested barrier to prevent everything that can damage the spermatogenic cell and support all nutrients and it produces the one protein called androgen binding protein to bind with the testosterone. The testosterone are secreted from the Leydig cell that locate out of this tubule, right? And testosterone sent into the tubule and bind with the androgen binding protein to work within the tubule. Okay. So the all function of the supporting cell or Sertoli cell is like a nurse, okay, like a nurse. And it also helps to phagocyte growth. The dead cell or the residual body is something shaping between the chain of the brazolytic to be the elongated spermatid. Okay. And help to facilitate the releasing of the sperm into the lumen by the contracted cell. Okay. And also secrete the suitable fluid for the sperm. And is also secreted inhibin. The inhibin function as the negative feedback for the sperm production. And this slide shows all of the spermatogenic cell containing the spermatogonia. And here primary and secondary spermatocyte, two types of the spermatocytes, and the round and elongate spermatid, two types of the spermatid. Okay. When we would like to simulate the subtype of spermatogonium, the first one, the spermatogonium type A occurs is differentiated from the stem cell. And when the spermatogonia type A differentiate to be the dark and pale subtype of spermatogonia. The dark spermatogonia is remain to be the stem cell here, but the pale spermatogonia will further differentiate into the type B, and type B differentiate in the mitotic division to be the primary spermatocyte. We just learned this detail only in the lecture, not in lab. Okay? In lab, we just find the spermatogonia is okay. Okay. And when the spermatogonia differentiate in the mitotic division to be the primary spermatocyte, and primary spermatocyte contain the spermatogonia contain 46 chromosome on 2n, the haploid chromosome. Diploid chromosome. When the mitotic division, the two daughter cells contain similar to the parental cell. So the primary spermatocyte also contain 46 and 2n, like a parental cell. And after that, when the primary spermatocyte pass through the meiosis 1, but undergo the meiosis 1, is divide the chromosome in half. After the meiosis 1, the sperm called the secondary spermatocyte is just only contain one egg or haploid chromosome or 23 chromosome pairs. And then the secondary spermatocyte quickly differentiate in the state of meiosis 2 to be the spermatid. And start at the secondary spermatocyte until the sperm contain 1 egg and 23. And this slide shows the two types of spermatid. The spermatid contains round and elongate spermatid. The round spermatid or early spermatid. The shape of the sperm like only round nucleus, round cell, something like that. And after that, it moves to the spermiogenesis process. After spermiogenesis process, the shape of sperm changes to the elongate on late spermatid. And in this, you may contain the first step called the spermatogenesis, the step to produce sperm. And the last step is called the spermiogenesis, to change the morphology of sperm. And this slide shows the conclusion about the number of chromosomes and type of sperm. Okay, I will say again. Process 1, the spermatogonia, 2n on 46 chromosomes, divide into the meiosis process to be the type A and B. A dark, B main stem cell. A pale differentiate to be the B type B. Okay, and then type B differentiate into the meiosis to be the primary differentiate into the meiosis to primary spermatocyte, and primary spermatocyte go to the meiosis 1 to be the secondary spermatocyte. The chromosome increase to the 23 and 1. And the secondary spermatocyte pass through the meiosis 2 to the spermatid. brown spermatic first, then brown spermatic brown spermatic change the morphology, and this process called the spermiogenesis to be delayed or elongate spermatic. And then the process for release sperm that called the spermiation is the release sperm process. The release sperm process comes from the contraction of myoid cell and contraction of steroid cell, which use both of them to release the sperm for the spermiation process. This process requires myoid cell and steroid cell to release sperm into the lumen. This slide shows the state of spermiogenesis. The first step called the Golgi phase, second cap phase, a chromosomal phase, and the last one is the maturation phase. The first phase, you can see the first here. Okay. The first phase, you can see form the vesicle containing the enzyme neurotypes of enzyme called the chromosomal vesicle. On this slide, so your chromosomal vesicle is the same. Okay. And this chromosomal vesicle contains numerous enzymes, and it's moved to the one side. I will call this side head of sperm. Okay. Move to the one side, and the centrosome start to elongate or fracture on first. Okay. And the cafe, you can see the chromosomal vesicle look like a cap on the head of sperm. Nucleus more elongated or more slender, and elongate or fetulum also occur. And the chromosomal phase or first phase, the rearrangement of mitochondria, you can see this. And the last one, the fully morphology of sperm here. And some cytoplasm will be shaded, and the sterile cell will be eaten like in the phagocytosed by sterile cell. Actually, I conclude all steps here. The flattening of nucleus, the formation of a chromosomal vesicle, growth of the fetulum occur, reorganization of the mitochondria, and shifting of unneeded cytoplasm and cellular body, and is phagocytosed by the sterile cell. Is all of the morphological change of the sperm in the spermatogenesis process. You just remember this slide is okay, and I don't want you to remember about the deeper details, something like that. Just understand it. Okay, I already told you about the spermiation that require the sterile cell and myoid cell. Myoid and sterile cell. Release the sperm into the lumen. And this slide shows the cross section in EM. And at the midpiece, you can see the numerous mitochondria around the fratulence. So when you cut the midpiece, you can see here mitochondria located around the fratulence here. Okay. When you cut the principal piece here, you can see the fibrous sheath. Thicker fibrous sheet located around, but the mitochondria are disappear. And at the tail or the end piece, just only thin the plasma membrane and thin fibrous sheet located here. we move to the outer of the seminuclear tube, the Leydig cell, that only one cell that we have to learn in the interstitial space. This is the Leydig cell. You just see the nucleus and the clearly nuclear mass. Okay. This one is the Leydig cell 2, and this one 2, and this one 2. Okay. The main function of Leydig cell secrete the testosterone. Okay. And the function of Leydig cell in this state of life, in embryo, it helps to develop the male genital organ. Okay. Like develop the penis, scrotum, testes, something like that. When in our body contain a higher rate of testosterone than estrogen and progesterone in female. And in puberty, the hormone peak again and help to secrete the sperm, like a sperm production, and also help to change the secondary sex characteristic. It changes your bodies and your emotion, your mood, the growth of hair in some area, like a change in the voice, a deeper voice, something like that, and increase the bone, increase the muscle mass, something like that. It's called the secondary sex characteristic. And in adult, this hormone just maintain the state of spermatogenesis process throughout the midlife. And sometimes the researcher they found the crystal of Leydig located in the cytoplasm of this cell. But this crystal also unknown function. But they found numerous linkages in the tumor when the man contained the Leydig cell tumor. The increased level of linkage of crystal have been found. Okay, this slide shows the neural control of men. The first, the hypothalamus, release the gonadotropin releasing hormone or GnRH from the hypothalamus, and the hypothalamus or GnRH induce releasing some hormone from the anterior pituitary gland, FSH and LH. Hormone FSH move to the seminal or seminiferous tube. It functions directly to control the steroid cell to secrete the ADP or androgen binding protein. LS is go to the interstitial space between the Leydig cell to secrete the testosterone. And then testosterone move to the seminiferous tube and bind with the androgen binding protein as a complex and induce the normal rate of spermatogenesis. But in ultimately, the testosterone move to the plasma or blood circulation and exhale to control the secondary sex characteristic and everything about the man. And the testosterone is function as the negative feedback on the both anterior pituitary gland and hypothalamus blood circulation. But the steroid cell also increase its own negative feedback, inhibit negative feedback on only anterior pituitary gland. The semen contains just only the scrotum, but the testes remain located on the internal or like that in your body, your hip, something like that. And this is called a cryptorchidism. That means an undescended cryptophylism, they had an infertility because of not suitable temperature to high temperature prosper because the body contains 36.5 Celsius. After the sperm secrete on the lumen of seminiferous tubule, it moved to the first tubuli recti located here. Okay, then the short tubule. When we would like to find the tubuli recti, we have to find the transition zone like this picture. This part is the seminiferous tubule part. You can see numerous cells of the spermatogenic cell, right? And the terminal part of seminiferous tubule, you just only see the circular cell only. Okay. And then the cell just only contain the simple cuboid. And this part is part of the tubuli recti. Tubuli recti or tubular rectus is okay. It's quite hard to identify this tubule in the flesh. And then when sperm moves to the tubular recti, it moves further to the lateral testis here. Okay. The lateral testis located within the mediastinum, and this area is a thicker part of tunica albuginea called the area mediastinum. But the tubule within mediastinum is called the lateral testis. The lateral testis lie with a simple cuboid or low columnar epithelium here. And then sperm move to this area. Efferent ductus or ductal line, efferent of the lateral testis. The efferent ductus located here. When we cut the epididymis, maybe it's you can see the efferent ductus together in the same slice. Okay, and this slide shows the distinction between the efferent ductus and epididymis. The efferent ductus is a connection between the testis and epididymis. This contains pseudo-certified columnar epithelium. And pseudo-certified columnar epithelium contains quite equal number of tall columnar cells and short cells. So you can see like a sawtooth because the two sides of cells get together. And when the sperm move from the efferent ductus or ductal efferent to the epididymis, you can see the epididymis also lie with the pseudo-certified columnar epithelium. But normally, the most of epithelium, you can see the tall principal cell and the small short basal cell. Short basal cell just located closely to the basement membrane, and all of them in the tall principal cell. And this cell contains the stereocilia here. You can see clearly about the stereocilia, okay, in the epididymis. Oh, sorry. And because of the epididymis are divided into head, body, and tail. Okay. This one couple that means head, corpus, body, and chorda is the tail of the epididymis. Okay. The smaller or shorter of cell from the caput to chorda or head to tail. Okay. But the same type of the epithelium. Just the shorter in the tail of the epithelium that you already see in the stereocilia, both of them. the main function of epididymis, the sperm form testis, called the immotile sperm from testis. When the sperm passes through the epididymis, the epididymis gives the energy for sperm movement. When sperm passes through epididymis, it's motile sperm. The major function is the maturation area of sperm. The immotile sperm or the immature sperm. Then after out of the exit, the epididymis will pass through the vas deferens. First area here, the vas deferens. So the long tube and help move sperm into the internal area. And this one is the bladder here. Okay, the vas deferens pass through the posterior of the bladder. So there are three thickest muscle, smooth muscle within this tube. Inner longitudinal here, inner longitudinal here, middle circular here, and outer longitudinal here. The thickest of the smooth muscle is located within this tube. Okay. Inner law, middle cell, and outer law. And the epithelium line with the pseudo-notified columnar epithelium. Okay. And the terminal part of the vas deferens, you can see two different points. The first point, the numerous fold of epithelium within the ampulla part or terminal part of vas. The terminal part of vas called the ampulla vas deferens. And one layer of muscle is disappeared. The inner longitudinal. You just only see the inner cell and outer law. And innermost layer is disappeared. Okay. When after sperm exit the ampulla part or terminal part of vas deferens, enter the ejaculatory duct, and this duct located within the prostate gland. You can see ejaculatory duct in the slide of prostate gland. Okay. The ejaculatory duct line with the simple columnar epithelium. If you cannot find this duct, it's okay. Okay, we don't focus that too much. Okay. The function of the duct helps to ejection of sperm out. Okay. After something like that. Okay, we move to the accessory gland. The two main glands that we have to learn today is seminal and prostate gland. Seminal and prostate gland. This picture shows the posterior part of the bladder is called the urinary bladder. Okay. The vas deferens move from the scrotum and to enter the inguinal on the posterior side of urinary bladder. Side and light. Okay. Oh, sorry. Not this one. This one. This one is the vas deferens. This is ureter. Ureter for urine. Okay. The vas deferens and this area is the dilate part called the ampulla. Ampulla of vas. That is part of vas deferens. And then the sperm pass through this one, the ejaculatory part located in the prostate gland. This one, the prostate gland. Okay. And then the sperm has to be secreted into the prostatic urethra, membranous urethra, and penile urethra. Okay. The seminal vesicle located on the lateral side, nearly the angular part of blood difference here, two sides. Today we learn about the seminal vesicle is the one gland, the second gland, prostate gland. The last one is the bulbourethral gland. Okay. The seminal vesicle, you can see the numerous flow of pseudo-certified columnar like this picture. This one shows the numerous flow. And the thinner layer of smooth muscle here. Thinner. Okay. The most area of this gland is the lumen here. The major function of seminal vesicle is the main secretion of fluid. More than 70 to 85% of all semen from one time of injection. Okay, this comes from the seminal vesicle fluid. Okay, and numerous nutrient for sperm called fructose. Sperm require fructose too much. And the other gland, the prostate gland. You can see the prostate gland one the simple columnar to the pseudo-defined columnar epithelium. So you can see like a shape like here because they contain a numerous type of cell located on the epithelium. And one important thing that you can control this gland is prostate gland. The prostatic concretion or pulmonary aminosia. The remaining of calcium or everything within the gland. This can secrete the alkaline secretion. So the major function of prostate gland secrete the fluid to wash the urethra for sperm, preparing for the sperm, and fluid is suitable, provide suitable pH for sperm. Okay. And the order may be they may be get the prostatomegaly, the bigger of the prostate gland, and we might think about the secrete the urine, because it make the like a smaller tube, smaller prostatic urethra than normal. Quite hard to secrete the urine. And the prostatocarcinoma, that means the cancer on the prostate gland. General area that originated cancer located at the peripheral zone. Okay. When the doctor would like to detect the high risk of the prostatocarcinoma, they collect the blood and test about the level of prostatic or prostate-specific antigen or PSA. The normal level of this antigen 4 nanogram per ml, and higher or this level of antigen may be higher risk of prostatocarcinoma. But the PAP, normally the PAP is allowed to test the follow-up after know that okay, he got the prostatic carcinoma, just say the follow-up. But just say the list of carcinoma, they use PSA. And bulbourethral gland, we have not a slide about this gland, okay, so we just learn in the lecture. The bulbourethral gland located in the membranous urethra here. is located around the membrana urethra, located within the urogenital diaphragm. Okay. And is it clear for lubrication fluid during the sexual activity or sexual arousal? Something like that. And this slide shows the difference in the histology between them. Seminal vesicle, numerous flow or highly flow of lumen. The prostate gland, you will see the prostate contribution of corpora marcia between lumen. And lobourethral gland. But in that is not a lobourethral gland. So we just look at on two glands. And this slide shows the passageway again. First, sperm produced here at the testes in semi-nuclear tube, and they move to the tubercular lepicentis, ductal and epididymis, and then they move to the vas deferens and ampulla parts of blood, and then ejaculatory duct. And for the prostatic urethra, membranous and penile urethra. This is the passageway of sperm. Okay. The seminal fluid or semen, you know as in the semen, right? The major volume of seminal fluid comes from the seminal vesicle. Don't be serious about the percent, something like that. And when we have to test the list for infantile from one male, the doctor checks first sperm concentration. When one time of injection, they collect in the tube and test the sperm concentration and test the whole volume of semen. Okay. Whole volume of semen first and check the sperm concentration. And when they found sperm there, they will check the sperm morphology and sperm motility or sperm movement. Okay. They check 4, 8, 1, 2, 3, 4. The volume of semen first, second the sperm concentration, third the sperm motility, and sperm morphology. I will skip this line because I think it looks like a cross anatomy. Okay, not histology. Okay. And the penis, when we cut the penis, I'm not sure we have learned about the basic anatomy already. Okay. When we cut this one, right? In histology, we will see this one. They call vas cavernosum here that contains the erectile tissue. And the lower, like the mouse, the corpus spongios. And you will see, you can identify the penile or spongy urethra here. Okay. And this one shows the cross section of the penis. On the middle area is called the vessel, central artery. The blood enters through the central artery and then the vas released into the sinusoid in the corpus cavernosum. It will locate in the erectile tissue. It causes the erection of the sperm. And this one is the spongy urethra. The penis, when we cross section the penis, the epithelium of penis depends on the area of the. Okay. When we cross section nearly to the bladder, we find a transitional epithelium. when we cut at the prostatic urethra or membranous urethra or the origin of the penile urethra, we can see the pseudo-stratified columnar epithelium. And then the cell wall located on the epithelium decrease the size, stratified columnar, stratified cuboid, and the terminal end and the gland of penis. You can find this stratified squamous epithelium. That is not the slide or part of penis. We just, I'm not sure we have the slide of the penis or pelvis, something like that. And that slide, the corpus cavernosum, you will see like this. And corpus spongiosum like this. This is the slide that we have, the left bit. This slide shows the morphology of the erectile tissue. You can see the numerous smooth muscle cells and numerous blood. This red dot is a red blood cell. Red blood cell. Because it's the property of the erectile tissue. The filamentous red cell located within this tissue and the body can leak out within the sinusoid space and cause the vigor of your penis. Wow, I just used the one ounce. Okay, this slide shows, we just learned only in the lecture again. The gland of leader here. This is the epithelium of gland. Okay, locate a long length of penis and urethra. Is held for lubricant during the sexual arousal. And precordial or Tyson gallery within the pupil. Can you remember the pupil? The skin cover the glands of penis. Okay, deeper in the pupil, like located, it can secrete the smegma that's called the infection in vagina. Okay, the smegma. This type of gland is the sebaceous gland. When we secrete like a gland on the axilla, it's called the basement. Because it secretes both concentration or component of cell together with the fluid. Okay? Not only secrete the fluid, but some part of cell secrete together. Okay? And this slide shows again about the prostatic carcinoma. When we would like to test the high risk of this carcinoma, we test the PSA, prostate specific antigen. Okay. And when we got this carcinoma, you can find the pleomorphic cell. The pleomorphic that means abnormal of epithelium of the prostate gland. And normally we find just only single layer of the epithelium. And there is no secretion from this gland. Okay, if you would like to read more, I use this book as a reference. I will send the quick video for Top Lab tomorrow. Okay, finished. Have a questions? I don't have a question because I don't understand.